Hello everyone. Today we are going to discuss about prenatal diagnosis, amniocentesis. Prenatal diagnosis. Prenatal diagnosis or prenatal screening is a testing for diseases or conditions in a fetus or embryo before it is born. Perinatology is a branch of medicine that is concerned with the well-being of fetus or a newborn infant, generally covering the period from about 26 weeks after fertilization to 4 weeks after birth. The main aim is to detect birth defects such as neural tube defects, Down syndrome, chromosome abnormalities, genetic diseases, and other conditions such as spina bifida, cleft palate, etc. Diagnostic prenatal testing can be invasive or non-invasive methods. An invasive method involves probes or needles being inserted into the uterus, such as amniocentesis, chorionic villus sampling, etc. The non-invasive techniques include examinations of the woman's womb through ultrasonography, CT, MRI, etc. There are mainly three purposes for prenatal diagnosis. They are to enable timely medical or surgical treatment of a condition before or after birth, to give the parents the chance to abort a fetus with the diagnosed condition, and to give parents the chance to prepare physiologically, psychologically, socially, financially, and medically for a baby with a health problem or disability, or for the likelihood of stillbirth. Amniocentesis. Amniocentesis involves taking a sample of amniotic fluid and the fetal cells from the sample and are grown and then analyzed for the presence of absence of the certain chromosomes, genes, or enzymes. It is usually done from the 14th to 18th week of pregnancy. During this stage, some fetal cells may appear floating in the amniotic fluid because there is a little amniotic fluid before the 14th week after LMNP. Amniocentesis is difficult to perform before this period. The volume of amniotic fluid is approximately 200 and 20 to 30 ml can be safely withdrawn. It is done by an experienced physician who is guided by ultrasonography for the outlining and positioning of the fetus and placenta. The amniotic fluid is collected by inserting a hollow needle through the mother's anterior abdominal and uterine walls into the amniotic cavity by piercing the amnion and chorion. A syringe is attached and the fluid is withdrawn. The cells in the fluid are cultured and multiplied in a suitable medium for further analysis. Amniocentesis is a common technique for detecting genetic diseases. More than 30 genetic disorders, including trisomies 13, 18, and 21, Turner's and Klinefelter syndromes, and familial multiple malformations, sickle cell anemia, etc., can be detected by amniocentesis. The karyotype analysis and banding pattern studies after amniocentesis will enable to detect the chromosomal anomalies and also determine the sex of the child. Thank you.